today what I had in mind is talking about this Python library it's actually for geospatial operations. So not normally like the type of data we work with, of course, but I'll sort of do this in two sections. So the first, I'll just go through the library itself and just like the general concepts of how it works. And then in the second half, I'll give like a motivating example. So like why are we talking about geospatial libraries today? So in short, basically, it turns out some of these functions are really useful for spatial transcriptomics in a recent problem I had solved. But yeah, we'll do a real world like motivating example in the second half. But yeah, so I'll just get started. So I think their documentation is really great. And I found this like a very straightforward package to learn. I thought it was really well designed. So they do have a nice page here for the introduction. So I'll just sort of start here. So I found it pretty useful. The general idea is that if you're familiar with like pandas, basically just like Python's library for manipulating stuff with data frames. It's almost like tidyverse-ish. But with pandas provides you like these data frames, just like a data frame in R. And GeoPandas extends that. Essentially, it's like a subclass of the pandas data frame. Right, it provides a subclass of the pandas data frame called a geo data frame. And it's basically just a pandas data frame with a special column called geometry. This geometry column is made up of a GeoPandas geo series, which is kind of like a pandas series, which is really just a column in a data frame. But then that has data about like, it can store data about points, about polygons, different types of geometries. And so all the other columns are just normal pandas series. So you can store like integers, booleans, whatever you want in the data section. So I guess briefly, like this index column in pandas, every data frame has an index, and an index helps you. It's kind of like row names in a data frame in R, but some special properties where you can sort of automatically merge columns with different sets of rows as long as the indices have some overlapping matches. So, but basically an index is just like the row names. And so basically, yeah, it's basically just a data frame with an extra geometry column that has data about shapes and stuff. So not too much added if you're familiar with pandas data frames. So I think you can have multiple geometry columns, but there's only one active geometry column. That's what they're mentioning here, because that allows you to perform certain sets of operations we'll get into that a little bit later, but like you can only really have one column that does that. So they provide some functions for reading files. So they mentioned a few different things. The one I had used actually was GeoJSON files, but they support a few different ones for reading files. So they have like the read_file function. That's pretty standard, right? So we'll go through this example data set, basically districts in New York City. So that they import this data set here, basically. So it has stuff like Staten Island, Queens, Brooklyn. And so it has different types of information, other just ordinary columns here. And then it has the geometry column, which is a multi-polygon object. I'm actually not sure what this means. I think this is maybe overlapping polygons, but because I've worked with like individual polygons. But anyways, it's a type of shape object basically that you can do various operations on. Okay, so yeah, you just do to file to write it to a GeoJSON, for example. It provides some methods that are really straightforward to use, where for sort of basic types of information that you might want to know about your geometries. So basically right here, like if you just want to know the area of the polygon or whatever shape you have in the geometry column, you just do the data frame dot area, and then you can append that to a new column called area. And these operations, in my experience, were like surprisingly fast. I mean, area, I guess, is not super complicated, but other things have been, it seems pretty well optimized in general. So that was cool to see. And then, so yeah, area is one of them. Other things like finding the centroid as well, very standard things you might want to do with shapes. Boundary, I'm not actually, this is, I'm less familiar with, because this is I actually learned of this library very recently to solve a very specific problem. So some of this is new to me. But yeah, I think boundary is just sort of the outside like polygon type of line string, I guess, object. So yeah, you can apply these basic methods, area, centroid, on your geometry, and then get those pretty quickly, add them to a column if you want to. Okay, so they show the centroid example. Okay, so there's this distance method as well. So if you want to find like the distance from the centroids, so you can find the center of each of these districts in New York City. So they use Staten Island as the reference, using this ILOC 0, which is saying like take the first value of the centroid column, and then let's find the distance to that centroid for every basically every value in the centroid column. So then that returns basically this new column where you have distances to Staten Island of each of the districts. So yeah, this distance method can be useful for certain things. They make a point here about saying that like, okay, geodata frames are just data frame, like a subclass of data frames. So they inherit all the methods and stuff. So you can do sort of normal, if you're, again, these sort of require you to be familiar with pandas a bit, but you can like dot mean as a method that you can call on pandas series. And so anything that you can do in pandas, you can do in GeoPandas is the idea, really. Now we get into plotting. So the nice thing about plotting in GeoPandas is it does sort of build off of matplotlib. So sort of the very standard Python plotting is supported. And so plotting is very simple the commands are very short and succinct, and they are pretty powerful. If you just do dot plot on your geodata frame, the first variable is what you color by. So we had computed the areas of each of these as a continuous variable. And so this is plotting the active geometry column, which has our polygons representing the districts, right? And then colored by the area of the district. So the largest district here is going to be yellow on the Viridis scale. And yeah, this is all ultimately using matplotlib. So if you're familiar with matplotlib, then you can do all the functions there, and it just works out of the box. So they have other functions. Like this one's really cool. It's sort of more designed for notebooks, which I don't use as frequently, but in like a Jupyter notebook or something, you can have this interactive map that you call using the explorer function. So it's just like dot plot, but you get an interactive map. I zoomed out really far out here. It was supposed to be, normally if you just load it, it would look something like this, right? So this is really cool. It already comes with basically some map data built in, and then plotting the positions of the polygons on top of the actual real New York City map. So this is pretty cool. And colored by, again, the area. And then you get some information that you get just from hovering over each of these regions or each of these districts, which provides actually information from each column in the geodata frame. So yeah, this is a really cool feature in like notebooks and stuff. I'm not sure how we would use it in like sort of our workflow where we usually submit stuff to a cluster, but yeah, this is pretty cool. So they show other types of like if for some reason you wanted to, you can set the active geometry column to, instead of to geometry like it was before, which contained our polygons. This is the centroid column where we had computed where the centroids are of each of the polygons. But so I mean, if for some reason, I'm not sure why they did this honestly, but this would be what you return when you use the plot function, right? So you just get the centroids down plotted, since that's the active geometry column, again colored by the areas. So you do see like differences in the areas, even though the The regions aren't actually shown in this plot, but yeah. So here they're doing some tricks with matplotlib. So if you have there's something called axis in matplotlib, and you can plot multiple things on them. So in this case, they're plotting the geometry column, and then which has the whole shape, and then the centroids on top of each other, and all in one plot. So the centroids, you can see it did work correctly. And then there's some fancier operations here. So there's something called a convex hull, right? So that's like you take a polygon and then you make it convex, meaning like between any 2 points, all points between those 2 points are within the polygon if it's defined as convex. But in any case, I'm not sure when we would use this, but there's some fancier operation space that you can do with these geometries. So yeah, you see these are now convex polygons rather than sort of bending in, which were convex. But something particularly useful is that you can use what's called buffering. So on the active geometry, if you call the dot buffer function on the geodata frame, you could say you provide units, whatever the units are of your data. It basically draws a radius of that distance around whatever the geometry is. So if they're points, then you get these red-looking points here. So that's actually what they did here. So okay, actually, let me clarify here. So GDF.buffer here expands each of the polygons representing the districts. So that in this plot below, that would be the light blue regions that are now expanded outside of this. They also showed like the white boundary. So you have the white boundary, and then the GDF.buffer showed like this little buffer around the boundary. And then same thing, the centroids are just points. So points don't have any dimension to them. But if you apply dot buffer, all of a sudden now they're circles with a finite area, right? So that's what these red points are. And so yeah, the buffer function is pretty useful here. And let's see if this, let me just, yeah, we can go a little bit through this. There's some sort of like geometric relationship questions you can ask between different parts of your data frame. So you can ask if the buffered region, for example, intersects one of your, which of the buffered districts, so districts with a little bit of area added around them, intersects Brooklyn. So most of the regions are basically nearby Brooklyn. That would be one way of interpreting it. So you can do like dot intersects, you can do dot within. So there's a few different operations here. This is cool, but I think there's sort of a more interesting section that I'll get to that provides more information about spatial relationships between different polygons and stuff. One thing to mention here is that technically a geoseries is associated with what they call a coordinate reference system. So often there's a different ways you can define these. So they mention there are geographic coordinate reference systems. I'll be abbreviating as CRS from here on. So in the data, you'll see this code, EPSG 4326, and that's referring to a very standard geographic CRS based on latitude and longitude. So your data will be in units of latitude and longitude. So that's sort of defining, how should I say this, the area on basically on a sphere or an ellipsoid, like the surface of the Earth. But in order to do sort of meaningful operations with GeoPandas is sort of based on planar operations, so perfectly 2D operations. So you need to project, if your data is in latitude and longitude, you need to project it to a different CRS that's flat, essentially, where you flatten your coordinates and you have them in units of feet or meters or whatever. So they sort of mentioned this. You can set it to, this is setting it to the geographic CRS, which plotting it is just going to look the same because you're not distorting any information like this. I think for spatial transcriptomics stuff, we probably, this is not super relevant. That's why I'm kind of going quickly through it, but just keep in mind if you do use this library, that make sure that your CRS is something like none. That's what I did for a problem I was working on. Or just make sure if it's trying to use something that's in latitude and longitude, for example, that's not sensible for flat data. So yeah. So I guess just briefly sort of go back and mention that, like, I was wondering on like these maps and stuff, if you take like do the dot area, is it actually doing an area on like a fake surface of an ellipsoid or something? It's not, it's doing flat area, and you would project your coordinates down into a system where area, sort of an equal area projection that preserves areas. But anyway, that's a little bit maybe ahead of the scope of what we'll do in spatial transcriptomics, but I was just curious about that when I was working with this library. So yeah, projections. Next, we'll do spatial joins, and then that'll sort of cover everything that I wanted to talk about for today in terms of theory before we jump into an example. So let me go up here. Here's where things get a little bit more interesting so there's something called attribute joins and there's spatial joins. I'll be talking mostly about spatial joins. So a spatial join is where you take two geodata frames and then you ask the relationship, you merge them based on the geometries of each of the data frames. So they give an example here of like, I guess they have this, actually, let me find an example here with, basically you can ask, if you have two data sets, you can ask things like, do the geometries of the first data set intersect the second data set? How many are totally within another? Like you can ask if like a city is within a state, or I think they do this thing with groceries that I'll mostly skip over because I'll have a full example, real example of this with spatial transcriptomics data, which I think will sort of illustrate the point a little bit better here. But basically it involves, you use this S-join function on geodata frames, and then you can do like a left join, an inner join, or a right join, or sort of standard SQL joins. And then they have these predicates that determine what the spatial relationship is when you're combining them. So you can ask things like, do they intersect, contains, there's a within. I think they mentioned some more down here. Touches, crosses, overlaps. So there's a lot of powerful things you can do here. This was like super well optimized also. Like I was surprised how fast executed for a data set I was working with. And so, yeah, if there's like, I guess we could pause here, maybe if there's any questions so far about like the theory of things before we jump into a real example. But I know this is very new material and not stuff we're normally working with, right? But just to make sure people are following. If not, I can go through. Yeah, so I can just basically, you're probably, at this point, you're probably wondering like, why am I talking about geospatial data in Python, our stats session? So here's where this is sort of a motivating example of something I actually worked on. So, so with some Visium data where you get the image and the spatial transcriptomics, you can segment the images. And so 10x Genomics provides like a tool called Space Ranger. Space Ranger segment actually is the tool in the command. And so this segmentation data gives you cells in this GeoJSON file, which is like originally what motivated me to get into this GeoPandas library. So the task I was trying to solve was I was trying to find, we have these GeoJSON files and we have Visium spots, and we wanted to count how many nuclei were in each spot. So that was just generally the motivation here. And so I'll show sort of, now I'll actually dive into an example of how to use this library to sort of solve a problem like that. So let me, okay, so let me just jump onto the cluster. And I won't be using notebooks, I'll be using sort of an interactive Python terminal here. I'll load a library, or load a module that I use for many different Python and R related things for Visium HD. And I'll jump into Python. So I already have this script that has done a lot of this, and I'll try to describe sort of how it's using this GeoPandas library to do this. So I'll just load a bunch of libraries. So basically with the Visium data, you get like, you have, for this problem I'm describing, you have your spots. So I have the path with the positions for each spot. I have this path that gives you the dimensions of each spot and number of pixels. So like the diameter of the spot. And then I have this GeoJSON file for the segmentation data of the nuclei. Then I actually we want to plot this on the actual H&E image. So we use our image here. And that's basically these paths that I have up top. So I'll probably just skip some of this. So we have a data set with a certain number of samples. I did this as an array job over multiple samples. So let's just say we're working on one sample, our first sample. And so I'm just going to subset these paths to the first sample in our data set. So that's all this is doing. And then we can get to some of the more interesting geospatial stuff here. So I'm going to read in the tissue positions file. So let me move this up a bit. Basically, that just has your barcode for every row is a spot. You have your barcode, then you have basically the XY coordinates of the centroids of each of these spots. And so that's going to be our geometry. Basically, we're going to have these points. We're going to represent these as points. So what I'm going to do is I'm going to instantiate a geodata frame. I'm going to use this points from XY function to import the XY coordinates and then turn these into actual points with a geometry column. So now instead of a pandas data frame, we have a geodata frame. And so that's the same thing. We just have all the data from our previous data frame, and then we have this geometry column with our spots represented as points with the XY. But spots aren't points, right? They have a finite area. They're not zero dimensional like points are. So we want to actually add the spot diameter. So we're going to use the dot buffer function. So first I just need to load in this path, which has the information about the spot diameters. Then I'm going to use the spot GDF.buffer. And then the buffer we're doing everything in units of pixels here, the pixels on the full resolution H&E image. So we're going to buffer by a radius of the diameter divided by 2. And so now if we go to look at the data frame, it's not a point anymore, it's a polygon. It's actually a polygon that's approximating a circle. So it has a bunch of points, but it's basically a circle. Okay, so now we have our spots represented as in a geodata frame with the correct geometry. Let's go to our nuclei that are segmented. So I'm going to just use the read file to read in this GeoJSON file. And then the default, I had mentioned this thing about CRS. So when you just use read file, I think it infers that you're using like latitude, longitude map data, which is not what we're doing. We're using sort of flat data with coordinates that are already projected down to 2. They're already 2-dimensional, right? So I'm just going to say set the CRS to none and say like, yes, I know what I'm doing. And we'll take a look at what that looks like in a second. So this is pretty simple. This is just, it gives you your cells, right? We've segmented cells. So it has an ID for each of those cells. And then the geometry, the segmentations are also represented as polygons in this geometry column. So I'll show sort of I use some matplotlib code here to basically plot. I want to plot these together, right? So we have the spots and we have the nuclei. And I wanted to get a sense of whether those were just lined up correctly with the coordinate system. So I did this. I used dot plot on each of the geometry columns, and then turns out it's upside down relative to the image. So I just inverted the y-axis. And then those plots kind of look like, let me find one. This. So you have like each of your geometries. You have the spots in black and then the nuclear segmentations in blue. It's a very low res image because I didn't want to generate a giant image file just to show the overlap. But that's sort of like a sanity check you can do. I do this thing where I use like the total bounds function, which draws a rectangle around the geometries of the entire column. So basically a rectangle surrounds all of the spots. And then I want to just find like the center, basically the midpoint of that. So that's for plotting later. Also, I showed spots and nuclei, but it'd be useful to have the H&E image also. So I'm going to read that in right here. And while that reads, I'm going to basically, there's like, I wrote a little function to plot the image, the spots, and the nuclei all at once. So I'll sort of describe a little bit of that, but that's basically I subset the image. It turns out you have to flip it upside down, but I subset the image to a small box that's at the midpoint of the spots in the Visium grid. Same here, you're subsetting based on the on that small region. So, and then you just define your figure and axes in matplotlib. You do dot plot on each of the geometries, giving them different colors and stuff. And then you can just do like plot.imshow show and then save it as a figure. And so those plots actually, I won't run it because I've already run it, but this would be like, I'll show you what that looks like. So that's like these types of plots. So you have the H&E image, you have the nuclear boundaries in blue, and then you have the spot boundaries in black. And so this way you can sort of see where your geometries are on the image. So we haven't counted anything yet. Eventually we want to count how many nuclei are in each spot, but this is just showing what the geometries look like when you overlay them. So here's where the cool part, this is where we're actually solving the problem here. So it turns out you can count nuclei in a spot with a spatial join in GeoPandas. So this is really the bulk of the work done in one line. The rest was just visualization and importing stuff. So how do we do this? We take the nuclear data frame, the nuclei segmentation data frame, the spot geo data frame, and we do a right join. So what that means is that we want to keep all the spots in the spot data frame, and potentially we care about the spots, not the nuclei. And then we're using this predicate within. So that's saying the nuclei, which is the left, within the right, the spots. So I'll show what that looks like. See how quick that was? That was less than like half a second. And we have thousands of spots and thousands of polygons and stuff. So it's pretty fast. So what that looks like now is you basically have your geo data frame with your columns from the spot data frame with the geometry here. And the special part here is that each row now is a nucleus, and the index represents a spot. So you might have we have two rows that have this index of 0, meaning that there are two nuclei in the spot with ID 0. And then you have the index associated with the nucleus. And so what you can do is you can count basically how many indices you have or how many rows you have per spot, and that gives you the number of nuclei already. So I had originally made a mistake here because it gives you NaN columns, even when you don't have any. That's basically when you don't have any nuclei in any spot. So there's still one row when you have 0 nuclei per spot. So that threw me off at first, but basically, yeah, you can just do sort of you can drop NA values. You can, like I said, you can group by the index, right? The index gives you the spots, and then you can do a dot size saying like, how many rows do you have per group? In other words, per spot. And then you can do that with different predicates. So you can say like how many nuclei are within the spot, how many intersect. And I could even, I'll probably work on this and do things with centroids, spot centroids and stuff, since that's really built into GeoPandas. But yeah, once you do this spatial join, then you can, and you count the nuclei and add it to the spot data frame, then you can sort of, we can do one of these overlay plots again. And this is sort of a sanity check. Here I'm subsetting to, basically these two lines are saying take the first spot in the data frame where we have 5 counted nuclei, and then use this plotting function on that. So that'll give you something like this, where this was using the within predicate. So there are 5, this was a sanity check saying like, yes, we do indeed have 5 nuclei within this first spot here. There's one, if we were using the intersects predicate, we would get 6 here.
That's actually a false positive, no?
This one's a false positive, yeah, for sure. So some of them are, we have noticed some false positives based on the depending on the H&E image, the quality of the H&E image. Since this is clearly just a gap in the tissue, but so yeah, there's a lot more we could do with this, but once you count the nuclei, then you can just export it. So I hope this is sort of a motivating example of like, as we work more with like data, with high resolution data where we have segmentations and stuff, maybe we can use some of these operations for different tasks. This was a very special task, or a very like specific task, I guess, but I think this was cool. I found it interesting to learn, and hopefully this could be something we maybe use for certain tasks in the future. That's all I had for today. I don't know if there's questions. I know I rushed through some of this Python code, but yeah, I found this a cool library to work with. But